I would like to welcome our first speaker of the day, Dr. Bryan Charleston. Bryan is one of two that have joined us from the UK today, and we understand it's a large commitment to come for a half-day meeting, especially when you're flying on Canadian Airlines with delays and such, and we really appreciate the effort you guys made to join us today. Bryan is the Institute Director at the Pirbright Institute. In his role, he provides advice and expertise on the design of infectious disease challenge models for a wide range of pathogens in important agricultural species. His research group's efforts are focused on understanding the immune response to the foot and mouth disease virus in cattle and to develop novel vaccines. Welcome, Bryan.
Thanks very much for the invitation. Delighted to be here and give some information about foot and mouth disease. But there's a wider agenda here that Volker and Paul and Andrew and I have been discussing for a number of years now, I think, to try and bring Pirbright and VIDO together and working more closely. As we've just heard, these global challenges are not going to be addressed alone. And so I think organisations such as ours and VIDO can do a lot of good together, and collaboration is key to that. So it's very much worth the effort to travel over here to speak to you today. So I'm going to give you an overview of foot and mouth disease and some of the science and some of the international situation, and then colleagues will give more. Well, I haven't seen their talks, but they're going to give more explanation of the wider issues around foot and mouth disease control. So, foot and mouth disease virus. It's a very simple virus, a very small virus. It's just a protein shell. That's a X-ray diffraction crystal structure there, back from the late 1980s, and just has a single-stranded RNA molecule. Really simple virus. There are multiple serotypes, so acronyms here: A, O, Asia, C, and the South African territory viruses. You don't get cross-protection if you're immune to one of those against the others, and it's even worse than that. Some of those, within some of those serotypes, there's so much variation, that immunization with one strain in that serotype won't protect against all strains, and that's what the little colored markers mean, these diagrams to show the great diversity. So that's one level of complexity, and the other level of complexity is it infects multiple species, including wildlife in some occasions, particularly African buffalo. So if you want to make a vaccine against foot and mouth disease, you've got to make a lot of vaccines. In your portfolio, you probably need about 20 in an emergency bank so that you can cover any eventuality of what's spreading around the world. Now, I'm going to use a series of slides to give you some background about foot and mouth disease, and they actually come from, it's going to look, there's not a consistent style to those slides, and that's because they come from an e-learning package we've put together to allow people to understand foot and mouth disease and how to control it. And that e-learning package has been accessed by a number of people worldwide, and indeed we've had requests, which we're doing, to translate into Chinese and French. So there's a lot of people eager to understand about this disease and how to control it. So it causes serious production losses in countries where it's endemic. This is a massive global disease. Should have said one thing right from the start there at the previous slide. When I moved into foot and mouth disease research in the early 2000s, after the outbreak in the UK, the previous head of the laboratory, who was close to retiring, said to me, just remember one thing about foot and mouth disease. Things happen really quickly, really fast, faster than anything else, any other viruses. And so days matter with the control of foot and mouth disease, and hopefully that will come across when we start describing control measures. The disease is difficult to control without a highly effective veterinary service, and it's extremely contagious, rapid virus replication, massive virus release, spread by direct and indirect means, and multiple host species. And immunity is serotype and sometimes strain-specific, exactly as I said. Massive cost globally. The figure there, in current price, is over 10 billion the UK outbreak would have cost, with all direct costs and indirect costs. And Jonathan Rushton has estimated the ongoing annual cost of foot and mouth disease around the world is about $5 billion US. Diagnosis, critical. Early diagnosis, critical. I'm sure you'll hear more about that from Charles and others later. But the first thing is early recognition when it's introduced into free countries. Being able to spot that disease quickly is critical. And then the application of diagnostic tests. This means greater awareness of farmers and veterinarians, which is why we've developed this e-learning, which has been accessed by people both in endemic and free countries. And in endemically infected countries, there's a need to understand the cost of disease and its epidemiology so that you can actually justify and target those control measures. I've already said this, but just to reiterate, number of susceptible hosts, and the headline there on the right, sheep, pigs, water buffalo, goats, cattle, and the sort of natural host of foot and mouth disease. It doesn't cause disease, but spreads very rapidly in that population is the African buffalo, which adds another layer of complexity to controlling this disease in Africa. You have a wildlife host acting as a reservoir. Clinical signs. It's lesions on the foot and mouth, as already said, these vesicles that burst. It's associated with severe production loss, something you hear, well, it doesn't cause mortality. It causes very severe production loss. Animals that are milking may stop milking and not start milking again until they have another calf. Animals grow more slowly, and so the veterinary economist I know coined the phrase, no sophisticated agricultural industry can coexist with foot and mouth disease, which is why countries have spent a lot of time and effort to eradicate it. I've already said about the multiple serotypes. The incubation period, I've got a graphic in a moment to explain the timeline of foot and mouth disease and within an individual animal and how that's and that accelerated process. And it's not a zoonotic disease. It is purely through the disruption to food security is its main impact on the human population. Survival in animal products, a route that the virus could enter into a country. Fortunately, we know a lot about that. There's been some really detailed review papers produced about that, and the rigor mortis and the change in pH will largely destroy the virus. But the virus can survive in bone marrow and lymph nodes and certain organs. So these are the routes where animal products could get into the country, and very aware of smuggled products, for example, being a real threat if that material is then fed to or pigs in particular. So a little bit of the background of the pathogenesis of foot and mouth disease. Timeline in days down the bottom here. Things happen fast with foot and mouth disease. So clinical signs, if we infect animals, or we know when a farm gets infected in natural infection, you'll start seeing clinical signs in 3 to 4 days. And virus in blood. So within 2 days, you can start to get detectable virus in blood, then peaks about 5 days and then comes down very quickly. You can get virus that stays in the back of the throat, essentially, associated with the lymphoid tissue, tonsils, etc., in the back of the throat. And that virus can persist at a low level over a long period of time, the so-called persistent infection. We're not sure of the epidemiological significance of that infection in terms of maintaining an outbreak, but there are cases from years ago, introductions into, for example, Mexico, where one possible explanation is these persistently infected animals not showing clinical signs will cause infection. Persistently infected buffalo, African buffalo, will infect cattle. We do know that. But that piece is uncertain. What I've shown here that's quite important, and we've actually used this in the UK and advice to other countries, in the double-headed arrow there, you can detect virus in blood before the onset of clinical signs. The antibody response is also very rapid, so that's in our favour. So that this is why the viremia is controlled quite quickly, and this is why clinical lesions are controlled quite quickly. This rapid induction of a very high levels of antibody titer, and that also works with the vaccine. One of the real positives of FMD vaccination is it acts really quickly. That's just after a single inoculation. You don't need a prime and boost to get those high levels, and I'll show you more about that later. And then through some very extensive studies, doing one-to-one transmission events over time course, and we actually demonstrated that the window of transmission is actually quite narrow. So, and transmission occurs, so you also not only can detect virus in the blood and in other sites before the onset of clinical signs, you can also detect them before transmission. Now, we don't have easy ways to do that yet, but there's a window of opportunity there if you've got a herd surrounding an infected farm of screening those animals and seeing whether they are infected before they start transmitting. And then transmission is curtailed very quickly, more than likely because of the onset of the antibody response, which also you get a mucosal antibody response. A lot of criticism for the models of foot and mouth disease in the 2001 outbreak, and I don't want to enter into that debate. The models were fine. Those models, the same basic principles, those models are still being applied, have been applied in COVID. The models aren't the problem. The problem was the data was that whenever you could detect virus, not the long-term infection, but during the shedding and the viremia, the animals were infectious for that long period of time. And I think that's where we got overestimates from those models of the likelihood of adjacent premises being infected. So highly infectious. You've got an R0 value which has gone into ordinary language now in newspapers of how infectious a virus is. You know, it's the order of 1.2 to 1.5 for COVID, for example. Probably a minimum of 10 and could get up to 40 for foot and mouth disease. So for a short period of time, it's highly infectious, the amount of virus that's produced. And some work we've started, and this is detecting foot and mouth disease in the air, in both in our containment units and in some overseas sites. And so before clinical signs, so before the animals start transmitting, it may be possible to detect virus in the air. So thinking about mass sampling techniques to try and identify, importantly, that herds haven't become infected, aren't likely to start transmitting. So you wouldn't need to apply control measures to those farms. That's how we see the value of that. So I'm going to switch gear now a little bit. This is telling you a bit about the process of how the virus is replicating the animal and spreading. Let's assume now we've got an outbreak and we're trying to understand exactly what's going on that outbreak, happening in that outbreak, and who's infecting who and where the virus is likely to go. So a little bit of a complicated slide, but this is data we analysed retrospectively from 2001, the UK outbreak. And I think we all know now about variants, again through COVID, how these variants develop and emerge over time. And this is really what was shown way back in the early 2000s for foot and mouth disease. So you've got amino acid here numbered for the first farm, the virus collected from the first farm. That infected the next farm. One of those changes has become fixed. Then it goes to farm 3. And you've got 2 of those changes have now become fixed in the virus. And then on to 4. And you see the idea, the virus starts accumulating these small changes. And actually it's reasonably consistent between between each farm transmission of about 4 of those changes occurring at each time it transmits from farm to farm. And so this can be used in real time to monitor an outbreak. So we've invested quite heavily in latest sequencing technologies. So we can, as soon as we get an isolate, we can sequence the full genome of that isolate in 24 hours. And that's really important. So we can follow outbreaks. And what this diagram here is trying to show, and I'll walk you through it, is what happened in 2007, the 2007 outbreak in the UK. So we had the first infected premises, 1 IP, 1B, and up there at the top. And so these circles are the number of changes that have occurred from what we think was the original inoculum to the virus that was seen on that first farm. And then you can see that it's about the same number of changes as we measured retrospectively in 2001. So the IP1, as I'm showing there, then IP2 get a few changes, and then the next herd that was detected as clinical was IP3. And because we're doing this real-time sequencing of the virus, there are too many changes. There's a farm missing. So we went back to the field teams, there's a farm missing. And I won't go through the details, but they went back and found this IP5. And so that's really critical of relying on the conventional epidemiology, but overlaying the molecular epidemiology, and so that you can make sure you're seeing all of the farms that are infected, and so effectively controlling all of those farms. And I'll give you some more examples of since then where that's been used in anger in other outbreaks throughout the world. And here's the example, really. Korea had a very severe outbreak of foot and mouth disease, and they're still bringing it under control. And I won't go through this detailed diagram, but what essentially through this sequencing analysis, what we could show is that it wasn't one incursion into Korea that was then continuing to spread. Through that variation in the sequence, we could show that there was a continual incursion of different isolates over time that was promoting that disease in Korea. And so again, it gives you the indication of how do you identify all of those sources of the infection and bringing them under control. So part of preparedness is to have this sequencing technology available, which I'm sure you do, and then having that molecular tracing, so in real time you can monitor the field teams and assist them in improving the control of these diseases. I've got an even more recent example coming up later on. So skipping again onto another topic, this is about emergency vaccines and what is the process of producing an emergency vaccine and then actually deploying that emergency vaccine. It's quite a simple vaccine in many ways. The first the method was developed over 50 years ago. Cells in culture, BHK cells infected with the virus. The virus then replicates rapidly, and then through a series of inactivation steps, two inactivation steps, then there's some degree of purification required, and this is mainly to remove some of the products that would confuse an assay that could be used to tell whether an animal's been vaccinated or vaccinated and infected. And then that's stored in liquid nitrogen. And I'll explain to you why that has to be stored in liquid nitrogen as the antigen. The antigen, when it's required, is then formulated with an adjuvant, usually an oil adjuvant, and then can be deployed as the final vaccine. So if you look at the plans in the USA, they're looking at a time anywhere between 7 and 21 days from placing an order to delivery. And then back to what I was saying to you, the onset of immunity post-vaccination is 4 to 7 days. That's a long time in a foot and mouth disease outbreak. And talking to Tom last night, I think you've got a tighter timeline for this, but still, if you could find a way of deploying vaccine immediately, it would be advantageous. So the alternative to stamping out would be vaccination, and this is the small FMDV outbreak of foot and mouth disease in Holland. This is while the FMDV outbreak in the UK had started in 2001. So they had a head start. They knew that there was this O serotype causing disease in the UK, and were able to initiate that preparation of vaccine production. So their eradication strategy was to stamping out infected herds and preemptive culling of farms of a 1 kilometre radius around the infected farms. This is a publication from somebody in Holland who was involved in the control program. This is quite telling, I think, and I think this is what we all believe. There are capacity problems. Killing large number of animals and destroying them within very tight timelines is extremely challenging and labour intensive. And that's what they found actually in Holland, and a decision to apply emergency vaccine was taken there. And vaccination of all susceptible animals within a 2 kilometre zone around farms. And then they needed to extend that vaccination program, and with the idea that they would vaccinate and then kill at a later date. There's another conversation which I'll leave others to discuss. But in total, 26 farms became infected. But I think it's a success story. So if you had your vaccine ready to be deployed quickly, it's going to be quicker than a culling and destroying of the infected material process, and it can yield successful results. Do you vaccinate once or twice? So this is some work we did, and this is a tetravalent vaccine, 4-component vaccine, where we looked at this, and for each serotype, it's largely the same story. So I'll just concentrate on the O serotype at the top here. So all the groups were given a single inoculation at day 0, and exactly as I've showed with the infected animals, you get a very, very rapid induction within about 7 days of a protective neutralizing antibody response, which is what we see here. Then a group was given a boost at 1 month, 28 days, and you got a nice boost in the titer, but then it does actually fall off quite rapidly. But the groups that we left a little bit longer, we've largely sustained a good antibody response here. 3 months we vaccinated animals, good boost is maintained over a long period of time. And then here, a group boosted at 6 months. And I think these not so bad for this O serotype, but you can see here with this SAT2 serotype that the antibody titers are really dropping off in the 6-month group. So I think we would recommend a single boost, and then if required, you could go back in at 3 months as a control measure. Okay, let's talk a little bit about vaccines then. And this is I've already said that there's the current vaccine, which is essentially whole virus. That's then the RNA is inactivated by chemical treatment. It's then gone through a degree of purification, formulated an oral adjuvant, and that's what's provided now. So this is the FMDV structure I've shown you before, and the different colours are the 3 outer capsid proteins. And you can already see these pentamer structures, which you'll see more clearly in a moment. What I really want you to look at is this little region here. This is VP2, this interface here. And this is part of the life cycle of part of why this virus grows so quickly and infects so quickly, is that it can release its genome quickly. I'll start the video running. And so either small changes in pH or actually quite modest changes in temperature, the virus becomes unstable. And these pentamers, it disassembles into these pentamers. And that little space I told you there between VP2, that's where the unzipping starts. That's where the whole capsid comes together. This material is not protective. It is only that intact capsid is not protective. There's even products marketed of individual peptides, ideas for individual proteins. I think we all agree that you need that intact capsid. The original adenovirus vaccine that was taken forward by Plum Island was producing these capsids. I know that program's closed now, but that was the same idea. So that's the critical thing, is to produce that capsid. And the reason for that is that the B-cell epitopes on this surface, they're either at these interfaces, that's critical, so you don't induce a protective response, but also that very rapid B-cell response that we see, that is a signature of antigens where you've got repeating elements over the structure. And it's so the repeat of here to here to here. It's that repeating structure that really allows the antigen to engage with B cells and stimulate a rapid antibody response. So this is a vaccine that we've spent about 15 years developing. That sounds a long time, and that's partly because you haven't got to produce one vaccine. Back in 2013, we had a highly effective A serotype vaccine, and we've had to produce a suite of other vaccines along similar lines. And we've expressed these empty capsids in a baculovirus, in insect cells, using a baculovirus vector and then into insect cells. So no virus at all is used in that process. But what that's allowed us to do, and this is a high resolution image of that little space I showed you between the VP2 on that previous diagram. And so we can make, in this case, a single amino acid change. So instead of this highly charged molecule here that's ready to fall apart and that's important for the virus life cycle. Because we don't need the virus life cycle anymore, everything's been produced from recombinant technology in insect cells, we can actually make, in this example, a new covalent bond. So from a very unstable structure, we've now got structures that are stable at 56 degrees C for many hours and can be stored at room temperature for very long periods of time. So this is the way we're pushing forward with this to try and develop a new vaccine that would one, tolerate any breaks in the cold chain in endemic countries, but also could be ready to use, ready formulated for use in an emergency situation. And I do acknowledge because Gordon's here, but I would acknowledge it anyway, tremendous backing from DEFRA post-2001 to really get this technology off the ground. So the stabilized empty capsids, already said, improved storage characteristics, safe production, could produce it here. There's no live virus required, and so increasing the total production capacity globally. These production sites currently have to be high containment facilities. They're difficult to build, expensive to build, difficult to maintain. You don't need the virus, you just need the sequence. And you put it into the vector gene synthesis expression, and you start producing your vaccine. And then there are no non-structural proteins, so your diagnostics, your DIVA diagnostics are very robust. Okay, almost a complete change of gear now. For give you an idea of what's happening around the world, I think at Pirbright we're in a very fortunate position in that alongside the research programs literally in the same building and in adjacent labs is the World Reference Laboratory for Foot and Mouth Disease. We're also the National Reference Laboratory for Control of an Outbreak in the UK. But it's through this information gathering around the world is the major role, and we have been that in that role for over 60 years now. But the research groups sitting alongside the surveillance groups, I think, is critical for the development of new tools. The scientists doing the basic work really have to listen to the people who are involved in the disease control. What do they need? What improved tools do they need? And a lot of the things I've described have come out of those discussions about the molecular epidemiology and the potential for new vaccine candidates. So, and here are our key partners. It's Charles here, up on in Canada, and then around the world. And it's through maintaining this network we try and get an understanding of the situation of foot and mouth disease, understanding the strains, advising governments and advising the vaccine production companies about whether they need to produce new vaccines for outbreaks that are occurring. So this is foot and mouth disease in a nutshell. This is one of the latest maps produced by the reference laboratory at Pirbright. And these grey areas are the infected areas. Huge numbers of animals are infected every day with this disease. And of course, that burden of disease does act as a threat for those three countries. And what here is shown is a nice piece of work I think the reference laboratory and the network have done, is that there are these epidemiological pools. So by and large, the same types of viruses circulate in these pools, and they had a particular strains would circulate, but we're seeing that more and more broken down with globalization, all those same reasons why we had COVID: globalization, spread of viruses, movement of people, etc. And this is sort of some highlights, really an overview, and I'll go into some more detail in some of these of what we've seen over the last few years. Outbreaks in Russia, Kazakhstan, outbreaks in Indonesia, outbreaks in India and the United Arab Emirates of an O serotype there. And South Africa used to be free and is now really struggling to control these outbreaks, and that obviously has trade implications, which you'll hear more about, I'm sure. Namibia and Zambia, also an O outbreak. Traditionally, there wasn't a great deal of O infection in the south of America, but the south of Africa, sorry. But we're seeing outbreaks there. Then Eastern Mediterranean, we're seeing another O serotype here that's causing infections. Also in Turkey and Algeria, and we're seeing quite a significant increase in the number of cases in northern Africa. And then this one is really quite strange, is that a strain in Egypt that had based on molecular tracing, pretty sure had come from Venezuela. So understanding how that transmission occurred is going to be very important. And then marked quite rightly in red is this SAT2 isolate, which we're pretty sure by sequencing we can trace back to Ethiopia and has swept across the Middle East. And I'll show you some more detail around that. So here's an FMD endemic region. There's a lot of FMD, a lot of vaccine used. You get a new variant and it sweeps through those populations like they're naive. So really challenging. Talked about the increase in number of outbreaks in Tunisia and Algeria and Morocco, and then of concern to Europe. We're now getting closer to Europe. And increased trade between sub-Saharan Africa and North Africa is probably the reason for that. A new road being built around the west of the Sahara. And so we're seeing sub-Saharan strains now in Northern Africa. South America, I talked about this one a little bit. I won't dwell on this too much in the interest of time, but through molecular tracing, I already said, we're seeing some outbreaks in Egypt, which had only been seen previously in South America. And this is this SAT2X1V that I've described, and maybe I'll just skip on to the next slide. So we're pretty sure from the sequence it came out of Ethiopia and has swept across, Jordan into Iraq and into Turkey, and is moving into the borders of Europe again. So this is a real threat, and getting enough of a vaccine that will protect against this particular virus is a challenge. And the other example I want to give you, going back to that molecular tracing, and this is now on a whole region level. And so here are all the sequences. Hopefully you're familiar with these types of trees now and the changes that are occurring. 20 changes here, 22 there. Here's the isolates we've seen from Iraq. Here's the isolates we've seen from Jordan. There are too many changes there. So somewhere in the Middle East, and we don't know where it is yet, there is a foci of this virus. And the challenge is to try and find that and bring it under control. But the first thing is to make sure we've got enough of the right quality and type of vaccine to control this infection. And this again is just a key role of the World Reference Laboratories to try and understand, will the current vaccines protect against the strains circulating? So here are two vaccine strains that are available for SAT2s. SAT2s and the O serotypes, I maybe should have said this before, are really unstable. They're really difficult to make vaccines against. There's a whole range of stabilities we can see between serotypes and strains, and these are difficult. And so this SAT2 Eritrea, and I won't go into the meaning of this, but basically we're getting good cross-protection of the SAT2 Eritrea vaccinated animals against that new SAT2 variant, but that older SAT2 Zimbabwe strain is not giving such good protection. So the focus is on producing enough of this material, but it is a challenge to get enough material for those very large populations infected. So just to summarize, really, very much European perspective, I'm afraid, but I mean, that's our focus in our part of the world. What are the threats to mainland Europe? We've got these viruses circulating in Turkey, and this new SAT2 is potentially a real threat. We've got this rise of viruses north into North Africa, another potential threat. And this one is interesting. So this is a strain that's circulating extensively in Asia, and it's not close to the borders of Europe. But I'm reminded of what being told back in pre-2001 and the O serotype outbreak in the UK. For a number of years previously, the World Reference Laboratory had seen this virus that was spreading rapidly throughout Asia. So there are differences as well in how fit those viruses are and how well they spread. And this is a particularly transmissible virus. And so it is of a concern. And of course, this all then informs the emergency vaccine banks. Have you got vaccines in your bank that will protect against those viruses. And I think that's the real value of understanding what's happening globally and both for the benefit of countries that are infected and trying to bring the disease under control, but also in terms of vaccine bank. And then the one I've said previously, this loop from South America. Do I need to say all of this? A lot of risks from those viruses I've just shown on the previous slide. This is critical, this real-time exchange of lab and epidemiological data, maintaining this network, trying to get web-based dashboards, sequences, making this information available to everyone, so everyone's informed of what's happening. We've got people really trying to keep these networks ahead of the World Reference Laboratories in Malaysia at the moment, talking to the people in Southeast Asia network to try and really understand what's happening in that region. And all this information is available as we try and get the information publicly available as quickly as possible. Quarterly reports and annual reports. Here's the links here. Happy for the slides to be shared for people to pick up the links. And it's that openness about what's happening is critical for us all being able to control these diseases. Acknowledgements to all the funders and all the organisations that are supporting our work and all the teams at Bryan. Thank you very much.